This video is continuation of Unit 2. Today we're going to be looking at Living Metabolism Part 2, which is going to primarily be talking about enzymes. A couple things first. Remember, most enzymes are proteins. Remember, proteins have a three-dimensional structure, then that structure determines their function. Enzymes are very similar. The structure of an enzyme very much determines what it can do, just like the structure of a wrench determines what it's able to do. You don't use a wrench to cut paper. You use scissors to do that because scissors have a shape that is designed to cut things. A wrench is not able to do that. It will just tear or break the paper. It's not good at that. And so in biological systems, enzymes work as a catalyst. What do we mean by that? Well, a catalyst is a substance that lowers the amount of energy that is necessary to begin the reaction. If you'll look at the black line in this graph, there's this amount of energy that's necessary to get the energy to be released. This is an exergonic reaction. And so energy is being released, but notice the amount of energy has to go up first before it can be released. And that energy is called the energy of activation. Energy of activation is the amount of energy necessary to start the chemical reaction. Well, if you'll look then at the red line is the same reaction, however, done with an enzyme. Notice the amount of energy that is necessary to begin that reaction is cut in half. The reaction still progresses to the same product and to the same point, but at a much lower energy expenditure. And so if you multiply this times thousands of reactions, then you have a lot lower energy need for the body. And so let's talk about enzymes a little bit more. So here's an enzyme, and this enzyme shows, or this picture shows you a couple of necessary words associated with enzymes. First, the word is the substrate. The substrate is the reactant in an enzyme reaction. This is the chemical or whatever that is going to be changed as a result of coming in contact with the enzyme. Typically, the enzyme will either take something and pull it apart or take two things and put them together. It could just be that the enzyme also just chemically rearranges the substrate in some way to make it different. Whatever be the case, that substrate is going to come away changed because of its contact with the enzyme. The enzyme itself, however, is not going to be changed. Go back to the scissors illustration. Take a pair of scissors and you cut some paper with it. The paper is the substrate. It's the thing that's been changed. That paper is now cut into two pieces. The scissors themselves have not been changed at all. They are able to do another reaction. And so that's how enzymes work as well. They change the substrate and then they're able to do another reaction, which again is a source of saving energy. The place the substrate reacts on the enzyme is called the active site. The active site is where the substrate is going to bind, and notice this active site is specifically designed for this little red thing here. It is a specific kind of shape. That protein shape determines its function. A couple things concerning the way that the substrate binds with the active site. If you'll notice in this picture where the substrate is kind of nestled in here, what happened to the active site? Well, the active site kind of closed around that substrate, and what this is called an induced fit. Think about when you put on a rubber glove. You take a rubber glove. That rubber glove is specifically shaped for a hand. Definitely a hand. 100% chance that there's going to be a hand in there. However, does it perfectly fit around your hand? No, you have to kind of squeeze your hand into there and it forms around your hand. And so that's an example of an induced fit. It's still designed for a hand, but that particular fit is going to kind of fit around the substrate. And when you have the substrate and the enzyme all together, that is called the enzyme substrate complex. Just fancy term for when two things are made up into one unit, that enzyme substrate complex. Here's another picture of that. Zoom out a little bit here. So this picture shows you that here's the two substrates. They come in, they bind to this enzyme. Again, notice that here's the active site here. Notice how the active site kind of squeezes onto the substrates, and when those substrates come away, they are completely changed. In this particular instance, you have two substrates go in, two substrates go out, but notice they are changed. They are different than when they first came in. The enzyme is unchanged. The substrates are the molecules that are changed. Enzymes perform optimally at certain conditions. If you think about the enzymes in our own bodies, they prefer the temperature 98.6. They don't operate well at other temperatures. As we get colder or warmer, those enzymes will start to malfunction. They also operate between a pH of 6 and 8. They don't like anything else. Our blood has a pH of 7.4. Anything less or more, the enzymes in the blood start to malfunction. And so all enzymes, no matter what living system we're talking about, have an optimal temperature or optimal pH or optimal amount of salinity or something that's going to cause them to work the best. I mean, you think about the enzymes in our stomach. There's a pH of 1 in that environment. Those enzymes operate best in that environment. They won't operate anywhere else. There are some other structures that can cause enzymes to not work the way that they should, and these structures are called inhibitors. There are different kinds of inhibitors for enzymes. There are some enzymes that are called competitive inhibitors. Here's a competitive inhibitor. Notice that there's the normal enzyme here, the normal substrate, active site. In this picture, you have this blue ball here that has got into the active site, and it's preventing the substrate from binding. It's called a competitive inhibitor because it's directly competing in the active site. It is not it's not anywhere else. It's binding directly to that active site, competing with the substrate, keeping the substrate from binding. Another type of inhibitor is a non-competitive inhibitor. So in this case, you have this little peach colored square that is bound to the back side of the enzyme, and that has caused the active site to change shape. That's preventing the substrate from binding. And so it's an inhibitor. It's still preventing the substrate from binding, but it's not competitive in that it's not competing directly with the active site. And so this graph here shows you how the rate of reaction changes based on the type of inhibitor that's introduced into the system. Normally when we're talking about reaction rate, we're looking at the slope of the line. So just simple math, rise over run. The steepest line is going to have the fastest rates or the best rates of reaction. The more slope, the less rate. And so look, the competitive inhibitor and the non-competitive inhibitor drastically change the rate of reaction. Sometimes the body uses this to its advantage. There are enzymes that have activators and inhibitors that both turn the enzyme on, activator, and turn the enzyme off, inhibitor, on purpose, keep them from working. Usually in this case, rather than binding with the active site, as you can see the active site of this particular enzyme, there are 4 active sites, you have this other site that's called an allosteric site, another place. And so this activator binds to the allosteric site that is turning the enzyme on. In this case, this inhibitor binds to this allosteric site and turns the enzyme off. And so the body can use this to control the rate of reaction with enzymes. And this is an important concept because this talks about ideas like homeostasis and others, and normally when we associate that, we talk about feedback inhibition, or what's called negative feedback. And this is the idea that the product of a reaction then turns off that reaction. If you think of homeostasis, like sweating, for instance, the Sweating is done because your body needs to cool down. Well, if your body gets hot, what is the product of that? The product of that is sweat. It's a process, right? Well, that sweat in turn cools you off, thus deactivating the need for sweating. It's a negative feedback. And so if you look at this picture, this particular chain of reactions is creating a product, and that product goes back and turns off the enzyme that started making the product. It's a self-regulating system to keep a reaction from getting out of hand. Now there are other types of reactions that are the opposite, where as the product causes the production of more product, and that would be a positive feedback system. Lastly, some types of enzymes work under this concept of cooperativity, where one active site helps or cooperates with another active site on the same molecule. How does this work? Well, red blood cells are an example of this. And basically what happens is when one substrate binds to the active site, it causes all the other active sites to need a substrate as well. And so if you think of this is a picture of a hemoglobin molecule, which is for carrying oxygen. If only one or two of those active sites were full of oxygen as the um, cells were gathering oxygen, it'd be very inefficient. And so this ensures that all the active sites are full, thus keeping this maximum distribution of oxygen throughout the blood. And so this is just one example of how the enzyme can increase efficiency in